This is a screen capture video to help explain the pH lab. There are several questions that students have about this, and I wanted to clarify some of those things. First of all, when you get ready to submit the lab, as you look at my cursor, I'm going back and forth across the page. These are the directions to the lab. I'll scroll down until you get to number 1. Please do not submit the directions with the lab. Just the questions, their answers, and the tables that you will be completing. And there's the table for activity A and the table for activity B. So the questions, answers, and the 2 tables are what you will be submitting. Just to tell you a little bit more about this, with the pH, it's a logarithmic scale. So when you change a whole number, say from 2 to 3, that's a 10-time or 10-fold change. Keep in mind that when you're talking about pure water, it has a neutral pH, and the hydroxide ions equal the hydrogen ions. Generally speaking, when we're talking about acids, we're talking about a solution that has a higher hydrogen ion concentration than hydroxide ions. If it's basic or alkaline, it'll have a higher hydroxide ion concentration than hydrogen ions. In the experiment, for the first few questions, 1 through 3, those involve the images that you should see. I'm going to pull these over where you can see them. You should be able to access an image that has the 3 tubes with solutions in it. For number 1, what color is the phenol red solution initially? That's this middle tube, and I put it in the middle so you could compare the other 2 to it versus if it was over to the left or right by itself. It'd be a little bit more difficult to make the comparison, especially between these 2. In number 2, what happens when hydrochloric acid is added? The tube on the left is the one that has the hydrochloric acid added to it. And then for number 3, the colors of the solution when somebody blows their breath or carbon dioxide into the solution, that's this tube over here on the right. So these tubes aren't necessarily in order 1 to 3. The tube that has nothing added to it, that's just the phenol red solution, is in the middle for a comparison. And then the tube that's being referred to for number 2 is on the left, and for number 3, that tube is on the far right. When you scroll on down through the directions and you look at the standards that were created, when you measure something, you have to have something to compare it to measure it, a known or an accepted. And so we have pH standards, solutions that were made up, and we know the pHs of those because they're labeled for you. They are in order from low pH to high pH. 2, 4, 6, 7, 8, 10, and 12. Remember, pH of 7 is neutral. And so by this image, you're able to connect in your mind a certain color with a pH. So it doesn't say on the tube, this is a pH of 2. You just understand when you see that bright red color, that's a pH of 2. When you see this dark green, however you want to describe that, that's a pH of 12. Okay, so this is our known, and you're going to write down the colors as best you can in this table. And it's somewhat subjective, so do the best you can on the colors when you're distinguishing the reds or the greens from each other. Then eventually, in the lab, we move from the knowns to the unknowns. And what you need for these, you need the rack of, sorry about that. I hit something that I shouldn't have. I'm going to pull this up and come over here. And the idea, I'm having trouble fitting these all in, but you get the idea as best I can. that these are labeled as to what substance they are, but we don't know the pH. So actually in this one photo, these are in the same order. The tubes in the back are kind of up higher. These are the same tubes you're looking at in this previous photo. So you don't actually have to have both images. You can just use this one image. You'll compare the color of the white grape juice to the closest color back here, and you will assign the pH of whatever color it's closest to. If you think it's between two colors, then let's say you think it's between 2 and 4, you might say it's 3. That's fine. Just do the best you can and go with the color of the solution and not any sediment that you see in the solution. Disregard the sediment, just the color of the solution. So you will assign pHs to all these home-based solutions. And you'll complete that table, and that's one of the things that you'll submit. There are some questions near the end to answer. Questions 4 through 7. Let's take a look at those. And for questions 4 through 7, that refers specifically to the graph. So keep in mind as you're looking at this graph, it starts at 0, not at the far left-hand side in the corner, which is what most of you are probably familiar with most graphs you see. This graph starts out here kind of in the middle at 0 milliliters of acid or base added. So this is the solution before any base or acid is added, and you can tell the pH by going up from the 0 and then looking at what pH that is. So the question is, what happens to the pH as acid is added? And that's referring to the part of the experiment that is described above. I'll let you read that on your own. But as you add acid, you start at 0, you add acid, and you watch what happens, and you'll describe that. What happens to the pH as base is added? Well, as you add base, see what happens to the pH, and you can describe that. In number 7, why does the pH level out after the large increase or decrease? Okay, so you see it decreasing quite a bit, and then it changes less here, and as we'd add more and more, it would change less and less and less. And then number 8, what was the beginning pH? That goes back to here. If you have any questions, let me know and I'll try to help you out.